So hi, and happy you could join for this talk on experimental animal models of multiple system atrophy. So I am Wouter Peelaerts. I am a senior postdoctoral fellow in the lab of neurobiology and gene therapy of Professor Veerle Baekelandt at the KU Leuven in Belgium. So during this session, I will be focusing on experimental animal models, and I'll be emphasizing some of the work that we've been doing over the last years. And I'll try to explain how we've been using these, let's say, recent developments to build experimental models so that we can better understand MSA, and also importantly, to develop models so that we can test new therapies that I think are very much needed for multiple system atrophy. Okay, so first I'll just quickly introduce the Sinuclenopathies and the concept and the idea behind alpha-synuclein protein strains. And the reason for this is because we initially started working on Parkinson's disease, and we kind of segued into multiple system atrophy research because of some of the discoveries that we did. And so I'll just try to frame this a little bit and then go into more detail and talk a bit more about synuclein protein strains, how we are using these for MSA animal modeling, and then also finally how we believe the environment, and with that I mean the cellular environment in MSA brain are also important for disease development, and also in this case here, the development of these experimental animal models. So first, I think most of us are familiar with the Sinuclenopathies, and so this is basically a large group of heterogeneous brain disorders in which there is multiple system atrophy, there is Parkinson's disease, and dementia with Lewy bodies. And so what really brings together all of these diseases are these clumps of alpha-synuclein protein that you'll find whenever you go look inside the brain of a person with either Parkinson's, DLB, or multiple system atrophy. So these synuclein protein aggregates is really an important key feature that you'll see in each of these diseases. So the first time that this protein was described and discovered was almost 20 years ago by Professor Maria Grazia Spilantini, where you can see here an image of a person with Parkinson's disease. And you can see these pigmented neurons here, which are dopamine neurons that bear some of these inclusions, these synuclein deposits that we believe are so important. And so this was also described in the substantia nigra. This is also an area, a brain region that I will talk about more in some of the next slides. So these were also found in the substantia nigra. And also here in Parkinson's and multiple system atrophy, you'll find that these pigmented neurons start to disappear and degenerate over time, which will then lead to the typical symptoms of both diseases. And so what are these synuclein deposits, or what are these aggregates? And so these synuclein protein clumps are basically big piles of synuclein protein that you'll find inside brain cells in the brain. And so if you, for instance, make a high magnification image of such a deposit, you can actually see, for instance, here within this cell, almost kind of a hole here. And if you zoom in here in this panel and this panel here, you'll see this proteinaceous substance here, this granular matter almost, which is all proteinaceous material. And you can see here at the edges, some lipids accumulating and some other important organelles and aggregates that are here present within these inclusions. And if you look very closely, you can sometimes even see this kind of rod-shaped like structures. And this is so this is really an important focus, and this is something that we believe is important in the disease pathogenesis. So you can also extract these inclusions or these deposits. You can isolate them from a person's brain. And here you can see 4 different panels, which are pictures from, I think, 4 different people with multiple system atrophy, where the researchers here looked at the synuclein filaments that were extracted from these deposits. So for instance, here you can see such a filament here as well. And you can see that they are also decorated with these black dots, which are markers or antibodies that specifically bind with the protein alpha-synuclein. And basically what this means is that these fibrils or these long filamentous assemblies of protein is basically stacked alpha-synuclein protein stacked up on top of each other, and thereby they form these almost spaghetti-like filamentous-like structures. So these are the alpha-synuclein fibrils, and these are also the structures that we've been studying a lot and that we are now using for our experimental models. So there are many similarities, but also many differences between these different synucleinopathies. For instance, in the case of Parkinson's disease and dementia with Lewy bodies, you'll find these big inclusions with those fibrils within neurons. And so whenever that happens, we mostly refer to those as Lewy bodies. And here you can also see another image, a picture of how that looks like inside the brain. In the case of multiple system atrophy, however, these inclusions do not form in neurons, but instead they form in a different cell type called oligodendrocytes, and thereby they form these glial cytoplasmic inclusions, or GCIs. And so this is really unique and very specific for multiple system atrophy, and this is really something that you won't observe in any other type of synucleinopathy. And so because of this, we also believe that MSA is a primary oligodendrogliopathy, meaning that the synuclein aggregates that are depositing within these glial cells, within these oligodendrocytes, affect the function and the health of these oligodendrocytes. And since they are also responsible for producing the white matter and the myelin that is important for metabolism and trophic support of these neurons, you'll see over time that the myelin starts to demyelinate, starts to disappear. And with that also that these neurons will also become dysfunctional and start to degenerate. In the case of Parkinson's disease, for instance, you'll see these deposits here in neurons. And in that case, we speak about a primary neuropathy where the lesion is actually found within this other cell type, the neuron itself. So those are the synucleinopathies. And so I think this is now almost more than 10 years ago when we started studying this. And one of the reasons why we got into this is because there seems to be this one molecular event that seems to be very important because we find it in so many people with different diseases. But what we didn't really understand at the time is how one key protein or one molecular event appears to be so important in this incredible heterogeneous group of diseases. So we couldn't really reconcile the idea or the fact that and at one side, you have this big group of diseases with very different clinical syndromes and phenotypes. And then at the basis of this, at the, let's say, the molecular underpinning of the disease, there seems to be this one event. So this didn't really make sense to us. And so we started wondering how that could be possible or whether we weren't overlooking something. And so then we thought, maybe we have to look at these fibrils and look at these fibrils in more detail. And we wondered if it was possible that these fibrils would have different structures, different shapes, that they wouldn't just be the simple rods or filaments that we thought they were, but maybe that they had different structures and different types of conformations, and with that, different biological functions. And if they had any toxicity, also different types of pathological functions. And so to study this, we started working with the lab of Professor Melky, who will also be presenting here during these sessions. And in Professor Melky's lab, there was Luc Bousset, who started experimenting with these synuclein fibrils. And he almost literally isolated himself for several months, and this was actually pre-pandemic. So this really, I think this really shows his determination there. But he really disappeared for several months, and then he really tried to assemble these different types of fibrils within a test tube. And so he actually succeeded in this, and he was also able to show that these filaments, these fibrils, had very different structures and very different biochemical and biophysical features. And so next we then asked what would happen if we would, for instance, inject these in an animal model. And so at that time we were developing several models. And so the model that we used for this type of experiment was a rat model where we genetically engineered these animals to express the human version of the alpha-synuclein protein. And so we do this by injecting these viral vectors into the brain directly, together with either the type 1 fibril here or the type 2 fibril here, which we called fibrils and ribbons. So this one we here called ribbons because of its weird ribbon-like looking shape. And so by doing so, by injecting these two different types of fibrils, which we also call strains, we found that the fibrils that we injected, so the type 1 fibril, caused neurotoxicity to such an extent that you can actually see here these pigmented neurons of the substantia nigra disappearing. So these fibrils caused very strong neurotoxicity, whereas these ribbons did not do this. Instead, these ribbons were causing a unique sinuclein pathology, meaning that, again, when we looked inside of the brain of these animals, we could find sinuclein deposits in different cell types. But what was actually quite striking is that we also saw them in oligodendrocytes. And this was actually the first time that we realized that different strains could be at the basis of different diseases. So Parkinson's disease and multiple system atrophy, because now we were seeing features and phenotypes that were actually specific for a certain disease. And so after this, we also wondered if we could, for instance, isolate these fibrils from a patient brain and then study them in our animal model. And I'm sure this technique will also be explained in some of the other sessions or the other talks. But very quickly here, what we did here is isolate these sinuclein deposits from a patient brain, a person with, for instance, Parkinson's disease or multiple system atrophy. So really extract them and then put them in a test tube and amplify fragments of, for instance, these fibrils here with sinuclein monomers. And so by doing so, we actually generate new fibrils by seeding and templating with the material in this test tube. And so by replicating this and amplifying this many different times, we end up with brain-derived fibrils that are so homogeneous and so pure that we can inject them and study them in an animal model. And so that's exactly what we did. And we used, again, the same model as the model that I just showed you before. So we have, again, here these rats that were expressing the human form of the alpha-synuclein protein via this genetically engineered trick and viral vectors that we were using. And so we co-injected these fibrils or these strains that we got from the brain of a person with PD, MSA, and DLB. And then we looked at the pathology that would develop. So here you see some of these results. So this is actually one slide summarizing several years of research. But what you see here, for instance, is that whenever we inject PD strains or MSA strains or DLB strains, whenever we inject any of these fibrils in the animal's brain, you can see that in the case of multiple system atrophy, there's very extensive cell loss, neurotoxicity, but also if we look again here within the brain, when we look at the sinuclein pathology and sinuclein deposits, you can see that in the case of multiple system atrophy, the pathology is actually the most severe. So showing that amongst all of these different sinuclenopathies, that these fibrils derived from MSA patient brain are in fact the most toxic species. So as a quick summary here for this first part, what we showed is that sinuclein fibrils, so these rods, these big inclusions, the positive alpha-synuclein, that they can have different conformations, different structures, and with that they can cause different and unique pathologies in vivo. Also that they can be isolated from patient brain, and that those isolated fibrils also in turn cause unique pathology in vivo. So then finally, the third topic I just wanted to touch upon is how the environment in multiple system atrophy, we believe, is also very important. Because as I mentioned before, again, you have these interesting differences, but also similarities. And one of the interesting things we noticed was this cellular tropism. Because in our animal models, when we inject, let's say, the different types of fibrils, and when we inject them in our animal models, we usually get a certain a mixed phenotype. So in other words, if we inject, for instance, a Parkinson's disease fibril or strain within our animal model, or when we inject an MSA strain in our animal model, we do not necessarily get a 100% complete phenotype. In case of the PD strain, we get a PD-like phenotype. And in case of the MSA strain, we get a more aggressive phenotype, a more aggressive disease. But what we don't see are these typical widespread and abundant glial cytoplasmic inclusions that are so typical of multiple system atrophy. So we do see some parts of it, but it is not fully penetrant. And so there was an important study that then found an answer to this, let's say, peculiar finding. And this was done by the group of Virginia Lee several years ago, where they did many different experiments, but where they showed that you could, for instance, add these fibrils to a Petri dish containing either neurons, so isolated neurons within a Petri dish, or you could also add them to a Petri dish containing oligodendrocytes. And so these cells, these glial cells or these neurons were then either expressing human alpha-synuclein. And so when they added these fibrils to this particular environment, they could see that, for instance, for these neurons, you would end up with a neuronal strain. So these fibrils would then be added here. They would actually amplify within these cells. They would reproduce even within these cells, and you could extract them again. And so here you would end up with a neuronal strain. But in the case of the oligodendrocytes, something really odd was happening. So if they again took this first fibril here and added to the oligodendrocytes, let it incubate for several days, and then extract it again, they found that suddenly these fibrils were turned into a slightly different version of themselves that were much more infectious and much more toxic than the fibrils that were initially added to that particular culture. So it shows that the glial or the oligodendroglial environment has a certain capacity or a certain function by which they can actually generate new types of strains that are unique and more damaging. So we also then went back to our initial experiments where we had these ribbons and these fibrils. We now wondered what would happen if we would change the environment in which these two types of fibrils or these two types of strains would amplify and propagate. So instead of the initial model now, we have a mouse model that was now genetically engineered to express the human alpha-synuclein protein within oligodendrocytes. And so by doing so, we actually soon realized that these fibrils were now causing MSA-like features. So for instance, if you look here at the bottom panel of this slide, where we have our three experimental paradigms. So here we have our control animals. So these are the animals that were genetically engineered to express alpha-synuclein in oligodendrocytes. Here you can see the animals that were injected with the ribbons. And here you can see these animals that were injected with the fibrils. So remember that these fibrils were the most neurotoxic strain or the neurotoxic fibril that we found initially. And also here you can see this big hole, which is basically brain atrophy. So there's tremendous brain atrophy after several months of injecting these fibrils within these animals' brain. What you also see here is that these white spots here, which is the myelin, is also absent here in the condition where we injected the fibrils. So there's demyelination, there's loss of white matter together with brain atrophy. And so these are very typical features of multiple system atrophy. And so we did this whole range of experiments and we measured a lot of different things. And so this is actually all to show that the fibrils were, again, the most neurotoxic species where you have loss of dopamine producing cells in the substantia nigra, But also there's a loss of medium spiny neurons, or there's a loss of cells that express the dopamine receptor. And that's another important feature of multiple system atrophy, because this only happens in MSA. And this is also the reason why, for instance, levodopa is not as effective in MSA, whereas in Parkinson's disease, these type of cells actually do survive. And also here, these are just more quantifications. So then next in this model, we wanted to investigate the type of inclusions that were being formed. So we have this interesting environment and we have these strains, and now we were wondering, what are these strains doing within these oligodendrocytes? And so to further analyze this and characterize this, we started looking in human brain. So here are actually a controlled tissue from a person without any neurological symptoms. And you can see the brain of an MSA patient that I stained for this marker of alpha-synuclein pathology. So just indicating that you here have these glial cytoplasmic inclusions that are very widespread throughout the putamen here. So this is one method of analyzing this. And here you can see a second method of analyzing this, where we use fluorescent molecules, fluorescent markers, to label these aggregates in different ways. For instance, here on the left side, the left panel of the picture, you again see the same control image, the same individual that doesn't have any pathology. Here in the top, you see a marker that specifically binds with aggregated alpha-synuclein. And here you see a marker that just recognizes the alpha-synuclein protein itself. So if you look here in the brain of this MSA patient, you see again the alpha-synuclein protein, but also here, if you look underneath this layer of markers, you can also see the aggregates popping up. And you can also see here, for instance, that they nicely co-localize, so co-deposit, meaning that the alpha-synuclein protein is aggregating within these glial cells. So then next, we applied this method, this technique, to our mice that, again, were genetically engineered to express the human alpha-synuclein. And so what you see here is that these mice, again, are positive for this marker, human alpha-synuclein. But when you look, say, underneath the hood here, you can see that there's basically no positive staining. There's no aggregation of the alpha-synuclein protein. So if we zoom in there, at a really high magnification, we can find some dots and some inclusions subcellularly. But if you just compare this with the MSA brain, you can clearly see that there are differences here, and that the type of inclusion and the type of deposit that is being formed is not exactly the same. Now, if we then inject these ribbons and these fibrils, you again see here the alpha-synuclein protein within the brain of these animals. But now what you can also see is that these aggregates are now appearing. So by injecting these strains, these fibrils, they are now seeding the aggregation of aggregated alpha-synuclein deposits within these oligodendroglial cells. And that, we believe, is very important. The other cool trick that you can do with these fluorescent molecules is that they also bind in a specific way to different type of aggregates. And the fluorescence of these aggregates is actually an indication of the conformational state of the proteins that they bind with. So in other words, these fluorescent probes are giving us a sort of signature or an aggregate fingerprint that tells us something about the shape and the conformation and what these aggregates are actually doing within here, these oligodendrocytes. And even to make it more visible here, we did this for all of our different experimental conditions. So here, the gray dots are just the animals that are genetically engineered to express the human alpha-synuclein. Here we injected the ribbons, here we injected the fibrils. And so here, these dots are oligodendroglial inclusions, aggregates found within the brain of a person with multiple system atrophy. And so you can see the this signature here, the emission spectra of all of these different aggregates. And maybe this doesn't seem like a big change, but you can also analyze it slightly different. And then what you see is that there are actually quite big differences between these different conditions. So here again, if we just take our normal mice that are again expressing the human alpha-synuclein within these oligodendrocytes, we find here this big stretch of data points, which means that the aggregates within this model are very heterogeneous, and that they don't really have, let's say, one specific structure. Now, when we inject the fibrils or the ribbons, we can see that this, what we call a folding landscape of conformational states, that the folding landscape is suddenly reduced, becomes narrower. And for instance, here in the case of the ribbons, you can actually see how suddenly it gets really close to the type of aggregates that we find in the brain of a person with multiple system atrophy. And so again, we think this is really important, and also I think very exciting work, because it shows that we can now develop an experimental model that can be modified in such a way that we are getting closer and closer to the real clinical situation or the real clinical condition. And by such, we can again understand much better what is happening, but also again develop, for instance, therapy that are specifically aimed at, for instance, removing these inclusions and removing these aggregates from a person's brain. So then as a quick second summary here, we now think that the phenotype in multiple system atrophy is not only determined by the strain itself, but also by the cellular milieu that exists in the brain of a person with multiple system atrophy. And so that both are actually needed to come to the phenotype that eventually develops. And so, for instance, here with these alpha-synuclein protein fibrils or these synuclein protein strains, we can just develop an animal model with MSA-like features, such as these behavioral deficits, brain atrophy, neuronal loss, develop oligodendroglial inclusions that are specific to a certain disease. And for instance, here, what I didn't show is also that these animals showed significant central and peripheral inflammation, which we believe also plays an important role in the disease. And this leaves me with a lot of acknowledgements of people that also contributed to this work. Of course, the lab of Veerle Baekelandt, with everybody that was involved here in this work, the lab of Ronald Melky and Luc Bousset and Alexis Vagny, the lab of Peter Nilsson, that also helped us to set up the HFDA assay and the fluorescent analyses, and Maria Stefanova for the transgenesis, Patrik Brundin and the KU Leuven imaging core, and of course, all of the sponsors that are so much needed and also helped us to do this type of research. And then, of course, I would like to thank you, all of the listeners, for attending this talk.